Hi, my name is Clarice Risseeuw and I'll happily present our paper entitled Reactivate, Redirect and Rearrange, exploring the design space of direct interactions with Flavobacteria. I'm presenting this work on behalf of my lovely co-authors Holly McQuillan, Joana Martins and Elvin Karana from Materials Experience Lab. Human connectedness to nature is of great importance for both the environmental crisis as well as human well-being. Yet ironically, human-nature interactions are increasingly scarce. Biodesign holds the potential to restore this by facilitating interactions with microorganisms. Researchers highlight that such interactions can both have a positive effect on human well-being as well as increase our ecological awareness and sense of responsibility towards a natural world. Living microorganisms are, for example, embedded in interactive installations and ambient displays. In most of these, the interaction between the human and the microorganism is indirect. This means that the human input is translated into a specific stimuli that is known to affect the microorganisms. Such mediation helps tackle challenges such as accuracy, control, and biosafety of living systems. Recently, SIGCHI designers have been showing an increasing interest in exploring more direct interactions with microorganisms. These intimate experiences hold the potential for establishing reciprocal relationships towards a culture of life, instead of using microorganisms strictly as controlled media. Tapping into this potential of intimate engagements, our research contributes to the SIGCHI community by inspiring and outlining the possibilities of direct interactions with microorganisms. We do this through our explorations with Flavobacteria. But before we dive into that, let me first share our definition. Direct interactions with microorganisms refer to instances where humans act upon living artifacts through their own bodies, for example through touch or movement, thereby inputting signals to the microorganism in an unmediated way. In response, these microorganisms adapt their behavior, often resulting in changes in their living aesthetics, for example alterations in color, that are perceivable by humans in close proximity. So back to Flavobacteria. 2 years ago we introduced these bacteria as color-changing interactive medium to SIGCHI. Flavobacteria species, in particular Cellulophaga lithica, can organize themselves into so-called photonic crystals, which produces vivid colourations similar to butterfly wings. They show great variety in their form, texture and iridescent color, as well as in their response time, and are affected by direct human input. Our research is guided by our curiosity and fascination with these microorganisms. We have embraced a material-driven design approach to center our attention to the distinct characteristics of these microorganisms while exploring potential experiences and interactions. Additionally, we have adopted an organism-centered approach by carefully considering the organism's needs. We used photography and video making to capture the microbes' responses to the interactions with them. Additionally, I took notes on my experiences when directly interacting with them. This allowed us to record the nuances of my personal experiences, which ultimately supported our ideation on application concepts. Our design space starts with the basic architecture for Flavobacteria-based living artifacts. These artifacts consist of the habitat enclosure, the microorganisms and the growth medium. While considering the microorganism needs, designers can tune the properties of these components. One can, for example, change the color of the growth medium to highlight the microorganisms and in this way surface their livingness. Next, we present interaction primitives. The first category is a series of input mechanisms through which humans can directly affect Flavobacteria's living color. For each of these stimuli, we also present a range that is essential for Flavobacteria's livingness and color. The second category covers several examples of human input. These are grouped into body, liquid and airflow, touch and movement. Next, we distinguish three ways in which humans can interact with Flavobacteria's living color: reactivate, redirect, and rearrange. Through the first interaction mechanisms, humans can activate Flavobacteria's living color as their input triggers the cells to organize into these optical structures. Once the iridescence fades away, Flavobacteria's living color can be reactivated by once again establishing the necessary conditions. Through the second interaction mechanisms, Flavobacteria's living color can be redirected, as input triggers the bacteria to organize their cells in a different manner or expand their colony in a particular direction. Thirdly, humans can interact with Flavobacteria by rearranging their living color. As the input physically interferes with the optical structures of the bacteria, the cells will reorganize themselves over time. This results in the elimination of living color and later on surprising colourations. To illustrate how this design space can inspire a broad diversity of direct interactions, we selected 3 human inputs to be further explored in our study, to either reactivate, redirect or rearrange Flavobacteria's living color. In our first study, we reactivate Flavobacteria's living color by pressing. Dormant Flavobacteria were applied to a flexible lid, which was subsequently pressed onto the nutritious agar medium. Over time, a colorful colony appears. It expands until the channel in the surface, which was intentionally created to allow the living color to be reactivated by pressing to the lid once again. During the reactivation, I was scared to break the foil as I did not want to contaminate Flavo with my microbiome. Instead, I wanted them to thrive and create structural color to their fullest potential. The interaction was very satisfying as the agar immediately broke and I could see the bacteria touching the new area in the petri dish, ready to explore new territory. In our second study, we redirect Flavobacteria's living color by tilting. First, we tilt one large custom-made habitat, which we compare to a horizontally placed habitat. While the effect only became apparent after 6 days, the bacteria drastically changed the way they expanded their colony. In a second experiment, we flipped the habitat back after 6 days. Here we could see that the bacteria showed a similar response and were able to adjust their behavior again. During the first experiment, I felt tempted to interact with the bacteria again after seeing the subtle changes in their living aesthetics. So it was truly satisfying and exciting to flip the habitat the other way in the second experiment and direct the bacteria to more space and nutrients. It was sad to see the bacteria running out of space in this habitat. I would have loved to keep this interaction going, redirecting them again and again. Finally, in our third study, we rearranged Flavobacteria's living color through swiping. The color was instantly eliminated when swiping through the optical structures and returned surprisingly fast. The color reappeared in 20 minutes. Instead of the pointillistic texture, the color was very uniform and only visible from very specific angles. When interacting with Flavobacteria in a repeating manner, the cells reorganized persistently into optical structures, recoloring the swiped areas. However, at some point, some parts of the colony became less bright and made me feel worried and guilty. Had there been some leftover alcohol in the glove through which I was trying to keep them safe from other microbes? Was I exhausting them by interacting with them so frequently? Or were there simply not enough cells left at the moment to form structural color? Based on these 3 studies, we identified application concepts. Here, direct interactions with Flavobacteria invite humans to reflect on everyday practices, while building awareness and sensitivities towards Flavobacteria's temporality, aesthetics and needs in shared habitats. In our concept Flavo12, Flavobacteria are integrated into a dress, foregrounding the history between humans and their garments. The unique dynamic colourations increase emotional attachment, but also invite humans to embrace the integration of nature into their daily lives. On a larger scale, direct interactions with Flavobacteria could capture how humans interact with their surroundings. In our concept Flavotread, the pressure of people's footsteps through a public space activate Flavobacteria's living color. This brings a highly artistic element to public spaces and encourages humans to notice, acknowledge and reflect upon the temporal beauty that microbes offer. Finally, our concept Flavotempo taps into the potential of Flavobacteria to foster mindful interactions. Here, the slow changes in Flavobacteria's living aesthetics in response to tilt create a unique and intriguing visual representation of time passing. Finally, I would like to reflect on some of the challenges and opportunities of direct interactions with microorganisms for HCI. First of all, the diverse temporalities of microbes. Microorganisms show a great variety of temporal behavior, ranging from immediate to gradual responses. Understanding this temporality is crucial for designers of living artifacts, allowing them to design the temporal scale of the artifact, but also to consider aligning human microbe temporalities for care practices. Secondly, the complexity and unpredictability of microbial behavior. As we move further away from lab environments and from the mediation of input to regulate living systems, it becomes harder to understand microbes' responses. This is because multiple input mechanisms can be in play. But even in relatively controlled habitats, unexpected behavior may occur. For example, in our first attempt to explore the effect of tilt. Here we observe some very uncommon behavior by Flavobacteria, for which the cause is still unknown. The next aspect concerns biosafety. Especially in this case of direct interactions with microorganisms, safeguarding the microbes' well-being becomes challenging, as inputs to living systems is not regulated and could potentially harm the microorganisms. Besides maintaining a safe environment for the non-humans, designers should consider safety concerns for humans as well as for surrounding ecosystems. Integrating microorganisms in living artifacts also raises ethical concerns. In line with the proposed organism-centered approaches, we encourage designers to carefully consider microbial sensitivities in the design time as well as through the entire life cycle of the organisms. Whereas we often struggled to move further away from our human-centered approach of considering Flavobacteria as living medium, there was a strong sense of what could be described as kinship, caregiving and responsibility in our experiments. Finally, our study revealed specific concerns regarding microbial semantics in these direct interactions. For example, it might feel disruptive to touch the bacteria through a pliable membrane, which might restrain people from interacting with the microbes. But it's also possible that direct interactions cultivate feelings of disgust and would therefore result in decreased empathy. So, in conclusion, our work introduces a design space that covers the basic architecture, various input mechanisms and examples of human input. We present 3 studies which involve directly interacting with Flavobacteria through pressing, tilting and swiping. We elaborate on application concepts for building awareness and sensitivities towards these microorganisms. And finally, we discuss the complexities and implications of direct interactions with microbes for HCI. Through our work, we seek to support the development of living artifacts that cultivate a sense of kinship and care and make humans feel more connected towards the microbial world and the ecosystems we all belong to.